Hi there, and thanks for the kind introduction and the invitation. So, what I'm hoping to do today is to actually bridge the fantastic talks we've already heard this morning to what I am really anticipating is an extremely interesting talk we'll have right after lunch, I think, with our keynote. And so, I'm going to show you a bit of the data we recently had, we recently got in our lab, and put that into context with cancer evolution. And this all stems from a review we recently wrote, Charlie, Kevin, and I, where we describe cancer evolution as a process grounded in Darwinian selection, but discuss and make our point that Darwinian dynamics and Darwinian principles are not sufficient or not enough to recapitulate what we've seen. And we've already seen some wonderful insights of that in the previous talk, and I would like to elaborate a bit more on that. So I'm sorry to say that this will be slightly less relevant to the metastatic setting, but I do actually think there will be quite some interesting points in there also for metastasis. And so, as we all know, and as we've seen over the past few decades, cancer can be seen and can be analyzed through evolutionary principles. On the left, you have a sketch of what Darwin wrote in his journals and in his diaries when thinking about evolution of the different species on Earth. And on the right, we have a schematic of a particular case of branched evolution showing the similarities between these two processes. And we, particularly in our lab, we are very interested in cancer evolution and to assess the relevance of the way cancers do evolve in time and space in terms of their impact in the patient's outcome and what we can do to make use of these principles to come up with better therapeutic strategies. So that's why a few years back we started two massive efforts, two massive studies called TRACERX and PEACE, from which you've already probably seen quite a lot of data over the years. But just to briefly recapitulate, we have access to a massive cohort of samples coming from different tumors. We try to get them. We try to get samples from before treatment initiation, during treatment, and hopefully even after treatment with the PEACE studies where we can perform rapid research autopsies. And we have mostly in Charlie's lab focused on lung cancer, but you've seen Summer's work in kidney cancer and melanoma, and we have other models as well. So the idea of of these projects is to study evolution through space and time to better understand how tumors evolve and what we can do about that once we know the impact of tumor evolution in tumor progression. And so the aims of we have several aims for these projects, and Samara has already showed you some results for that. And I was extremely pleased to be talking right after a talk focusing on ctDNA and CTCs, which are extremely important. So we'll not get into that over this talk because I could not do a better job than our previous speaker. But I will try to discuss briefly the relationship between tumor evolution and the outcome we see in patients, whether we can predict some of these evolutionary routes and what we can do about them in terms of a therapeutic outcome. And so I will now try to discuss, starting from what we wrote in the review, as to what different scales of evolution means, and so what different levels of alterations we can see of different contributors in tumor that can impact its evolution. So from microscopic level, thinking about genetic alterations, to a macroscopic level where we discuss the modes of tumor evolution. And so I will not dwell too much on this because I think this is kind of the basic concept that it's that shapes the way we see cancer, which is gradualism and DNA mutation. So this concept is heavily grounded in Darwinian studies, where, as you can see here with the quote from Darwin, where we think about mutations as individual alterations that happen in cancer cells, and will and may have the potential to eventually promote tumor growth and progression. So Darwin, in his studies when thinking about evolution of the species, was making a claim that nature does not make any jumps, that this process is slow and continuous and gradual, and will bring about changes over time. However, when we were looking at the data we got from our TRACERX and PEACE studies, we immediately realized that the sheer number of single nucleotide alterations we could see in our tumors was not correlating with patient survival. So we wondered whether there was something else we needed to be looking at. And what immediately caught our attention was the fact that we could often, if not always, see macroevolutionary events, and not single nucleotide alterations, but we would see chromosomal aberration. We would see profound chromosomal instability, something that Samra has already showed you already. And this brought our attention to such changes. And we started to think about this in terms of how these changes can affect evolution. And when reading what Goldschmidt wrote decades ago now, is that while single nucleotide alteration allows cells to gradually evolve over time, macro alterations can bring about massive changes in a relatively short amount of time. So we wonder what these changes could do over the course of tumor evolution. And one of the first things we could see and measure, as you can see down here, is that when you have chromosomal instability or SCN, we can see that these do actually correlate with worse outcome. So the more SCN you have, the poorer the patient's outcome is. And also in another studies which we collaborated, you can see that genome instability and chromosomal instability is actually linked with metastasis, as you can see in the graph here on the right. You have primary and metastatic samples, and this is a mouse studies, but we can see also the same in human sample. You see that the more chromosomal instability you have in your tumor, the more likely this tumor will be metastatic tumor, and that the prognosis of these patients will be poorer, suggesting that actually these tumors may need to acquire, to develop chromosomal instability in order to acquire metastatic features. And another thing that we focused on was the consequence of this chromosomal instability. And one of these is somatic copy number alteration, SCNAs, which are a pervasive event in cancer. Here you can see a massive cohort of different tumor types, and where we can see most of them, virtually every single tumor having at least one SCNA. And importantly, we see a difference between the number of subclonal SCNAs and clonal SCNAs, indicating that there is difference in the time where these SCNAs are acquired, and we actually mostly see them in mid-late phases of tumor evolution, as Samra has shown you as well. And so when we look at these copy number alterations, what we see is that there seems to be a prevalence for selection of these alterations, so that these alterations are under positive selection, because when we look at what is either gained or lost, we see that it is what the segments that are often lost in tumor are predominantly have tumor suppressor genes, while they have rather few oncogenes, and the other way around is true as well. So if you gain an arm of a chromosome, you usually have less tumor suppressor genes and more oncogenes. However, as I said, whilst this process seems to be under selection because it's prevalent, there's prevalent selection, what we do also see it's a contribution of neutral evolution, and I will get to that in a few slides. And so when we looked even more closely at these alterations, we kind of recapitulated the results we got with chromosomal instability, because we see that copy number alterations are frequently enriched in metastasis. And importantly, we see that this some copy number alterations, several of them that we see as minor subclones in primary tumors then become dominant clones in metastasis, suggesting that these ones are the ones that have acquired the necessary features needed for metastatization and colonization of a different environment. And closely related to this, so chromosomal instability and copy number alteration is another concept broadly explored in the lab, which is whole genome doubling. And this is work from Nikki that came out last year, where we could see that most tumors are actually frequently amplifying or doubling their genome. And we have seen a correlation between the proportion. When we look at broadly different tumor types, we see a correlation between the proportion of all genome doubling events with loss of heterozygosity, suggesting that all genome doubling is an event that is needed to better tolerate loss of heterozygosity, because if you have more genomic material, if you have double the number of copies, you can start playing around with the pieces of those chromosomes that you can eventually lose or gain to evolve. And now a concept we haven't explored so much, but many others have, and which is another extremely important macroevolutionary events, are catastrophic events such as chromoplexy, chromothripsis, and breakage fusion bridge cycles. These are massive events where in just a few cell cycles, sometimes even in one cycle, you have a dramatic rearrangement of the chromosomal and the genomic makeup of a cell. You can see here different examples, and we and others have shown how these are extremely relevant in terms of cancer evolution, and we'll get in that. I will get to that in a few slides. But then, moving away from the genomic makeup of a cell, or at least the chromosomal makeup of a cell, what we also focused our attention on more recently, and there's a couple of people in the lab working on this, is extrachromosomal DNAs, which, as you know, are circularized fragments of DNA which are present, often found in the nuclei or in the nucleus of cancer cells, and these are actually not defying Darwinian genetics, but they are non-Mendelian in terms of their inheritance. So in these DNA copies you have in the circles of DNA, you usually have oncogenes, which are then amplified, as you can see on the right. You can frequently find them on extrachromosomal DNAs. We see them in most tumor types, at least 1 or 2 copies per cell. And importantly, these are example of discordant inheritance, because extrachromosomal DNA is not equally segregated as chromosomes usually are, even in cancer, but they can be segregated unequally, conferring to different cells, different copies of these extrachromosomal DNAs, which can further increase tumor heterogeneity. And then one additional aspect we need to think about, and this particular dear to my heart, is cell plasticity, because as we've heard before, we are mostly focusing on DNA and changes in the DNA makeup of a cell. However, more and more papers are showing how we need to also look at several layers above the DNA. So we need to look at epigenetics, and that's what Samra is focusing on right now, I think. And then transcriptomic changes as well. So we see that cancer cells, and we've known this for a long time with EMT, we know this in melanoma with phenotype switch, can actually differentiate and de-differentiate at will at times. And this is extremely important in terms of therapeutic pressure, because we and others have seen that when you expose cancer cells to therapy, some of them will undergo a reversible reprogramming to adapt to this new stressful condition, and they will form a reservoir of cells, usually called the minimal residual disease, where cells can wait out the therapy and try out novel strategies to acquire resistance to it. And then one last point I will make about this, and I will not get into details of this because it will be another talk. Actually, is a microenvironment, and as was indicated in the previous talks as well. We all know how important it is, and I'm not gonna get into that. But what I would like to stress the attention on is some key questions that keep bothering me and don't make me sleep at night, actually. And I do think that at the moment we are still underestimating the relevance of the microenvironment. I mean, we've seen beautiful work from James Gregory's lab where he clearly demonstrated the relevance of an aged soma and a weak immune system in the process of cancer formation. And we know that cancer arise, most cancers arise late into age, and this is not necessarily because of an accumulation of mutation over time, over the lifespan of an individual, but it showed that an aged soma and a weakened immune system can actually promote cancer formation. But what I also want to stress, or I would like to stress, is that healthy tissues often show the same kind of mutations you see in tumors. However, these tissues do not form cancers, and the same thing goes on for. And another thing that really interests me is that you see that several mutations that are we know predisposed to cancer only cause very specific cancer types. We know this for BRCA, we know this for BRAF, we know for several diseases which have very peculiar, very specific cancer types as a consequence. And also in a similar context, we know that some cancers, not all of them, but many of them do metastasize a specific location. So this must mean that there is something in the microenvironment, both at the primary location site and at the location of metastasis, which is driving cancer formation and promoting cancer growth. And so for the last few minutes of my talk, I would like to focus on to just give a bit of context in the different modes of evolution. And we and others have shown the relevance of branched evolution, and I'm pretty sure I don't need to get into the details of that. But the basis is that there are different branches that are stemming out from a trunk, which has a variable length, and there are several different clonal selection sweeps that shape the evolution of cancer. But what I've also showed you before with the processes like chromoplexy, chromothripsis, BFP, but also SCN, is that there are in some cases we have massive rearrangements in just a short span, and this was also shown by Samra, which are able to generate what Goldschmidt called awful monsters. So a cancer cell which is whose genome is completely rearranged in just a few divisions and creates a chimera, a monster. And these are punctuated events. And then lastly, I would also like to briefly mention the role of neutral evolution, which you have also seen when we were looking at our single somatic copy number alterations. And this concept suggests that some tumors do evolve in a neutral fashion where there is no apparent selection of any particular clone. So at the end, you have a plethora of clones, all of them equally represented within the tumor, suggesting indeed there is no selection. And this was shown by two studies a couple of years back, where they showed that some tumors are mostly neutral, some mostly non-neutral, and then there are mixed phenotypes. What I'd like to briefly mention, though, is that neutral evolution does not exclude selection, and vice versa, because we've seen and we keep on seeing animal species, and I'm thinking about the elephant shark over here or the coelacanth, which are considered living fossils because we know that they have been living for a long time and they've been evolving very little throughout the last 350 million years, and have virtually neutrally evolved in this time span. But what we know is that a static habitat and the lack of predation does not promote selection. And so if near optimal fitness is achieved, there is very little need to evolve any further. And as the authors of the neutral evolution studies have shown, is that, I've suggested is that the situation may change because when selective pressures and bottlenecks such as metastatization are introduced in the game, then these will change the rule of the game, and these non-neutral evolutionary patterns may emerge. I will conclude by showing just a few slides on the implications of tumor evolution for therapy and outcome, trying to link with the next speaker in the next keynote. And I will just show the striking difference between what we consider as conventional therapy, where we do give a drug, we see a response, we see a minimal residual disease remaining. And from these cells, then what we'll have likely is the acquisition of resistance, relapse, and patient's death. And we've seen this over and over and again in the last 10, 20 years with targeted therapy. We've seen this also with immune therapy, and mostly with targeted therapy. So what evolution can do to help, what an evolutionary framework could do to help patients' treatment. And I will not dwell too much on this because this will probably be the main subject of the next talk. But one way of thinking about this is rather than aiming for tumor eradication is to aim for tumor maintenance and control, whereby different on and off cycles of therapy can actually stabilize tumor growth. And although this idea was proven to be correct both in vitro and in animal studies and in small clinical trials, when these studies were then repeated in larger clinical trials, they failed to achieve the intended objectives. Though it is worth mentioning that even if they are achieving equally as good as they're performing as good as the standard of care, in this case you're using way less therapeutic agents, and you are also not reducing the adverse effects of the patients. And also what I'm excited about is that are these two clinical trials led also by the next speaker, and I'm wondering whether we will get to hear about these promising results in the next talk. And then I think for the sake of time, I will just briefly mention these two different these two other modalities. So collateral sensitivity is a concept whereby one tries to exploit the known mechanism of acquired resistance to then target that mechanism specifically and eradicate tumor cells. And also a similar approach, which is called double bind therapy, where one tries to convert to the first drug the tumor population in a particularly sensitive tumor population for a particular drug, and then lead to tumor elimination. We've seen promising results in vivo of both these techniques. And then the last point I'd like to make is that one of the approaches that I think will revolutionize this field is to prevent the adaptation, because we know that tumors need time to adapt to a particular therapy. Sometimes they have intrinsic resistance, but in many cases we see that they actually initially respond and then acquire resistance. And so if we try to prevent the acquisition of resistance, and we've seen this, we've seen resistant mechanism at the DNA level with increased mutational rates. If we target those mechanisms, or if we target non-genetic mechanism of acquired resistance, as was shown in several recent papers, we may be able to achieve tumor elimination. And with that, I hope I've convinced you that cancer has to be looked at through evolutionary lenses if we want to achieve our aim of eradicating it. And I'm particularly excited by the studies that Summers and the previous speakers have shown, where we can actually get better information, because for these evolutionary therapies, these evolutionary strategies to work, we need way more information, and multi-regional longitudinal biopsies, liquid biopsies, and rapid research autopsies are the way to go. We need to expand our view, our gene-centric view, to include cell plasticities, the TMA, the aging soma, in order to understand the common mechanisms employed by cancer cells in response to therapy to target those. And then we need to harness and integrate this information to have better models and to design better evolutionary-based treatments aimed to predict and adapt to such a dynamic system as cancer. And I'd like to conclude by thanking all the people that helped me with this, especially my supervisors, Gavin and Charlie, all the people in their lab. I'm only showing just a few. The TRACERX Consortium, the PEACE Consortium, but especially the patients and families, and our funding agencies, two of which are actually organizing this meeting. So I thank them particularly. And I'd like to thank you all for your attention, and I'll be more than happy to receive any questions.